Good morning. In today's presentation, we are going to see about the edible vaccines. This is according to the syllabus of 4th semester B.Pharm. So before going into the edible vaccine, we should know what do you mean by a vaccine. A vaccine is a biological preparation that improves immunity to a particular disease. We all very well know about the vaccine and the schedule of vaccine, what should be given to the children, and we have studied in detail. So this word vaccine is derived from the usage of cowpox. Cow means vacca in Latin. It contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbes, or otherwise its toxins or one of its surface proteins. That means in detail we would have studied about what does a vaccine consist of. The vaccine may contain the live microorganisms or attenuated microorganisms. What do you mean by attenuated? That means it is like half killed. That means its pathogenicity is retained. That is called as the attenuated vaccine, which contains the weakened or killed forms of the microbes. Or otherwise, they may contain their toxins, that is nothing but their metabolic waste products, maybe endotoxins or exotoxins, or one of its surface proteins. These vaccines are the agent which stimulates the body's immune system to recognize the foreign antigen. So once it recognizes the foreign antigen, it tries to produce the antibodies which fights against the foreign antigen and kills it. In 1796, a person named Edward Jenner used this vaccine in human beings for the treatment of smallpox. And later, Louis Pasteur carried out the Jenner's work and contributed a lot towards the immune system. A vaccine is a biological preparation which establishes or improves the immunity to a particular disease by the body. This vaccine may be a prophylactic, that is, which will prevent the effects of a future infection by any natural or wild pathogen. Or sometimes it may be therapeutic, that is, vaccine against the treatment of cancer. When you say a vaccine, it should have some ideal characters. So what are the ideal characters of vaccines? It should be non-toxic or non-pathogenic. It should have lower levels of side effects. It should not contaminate the environment. It should not cause problems in the individual other than producing the antibodies. The technique of vaccination should be simple, and also cost-wise it should be cheap. So these are all the various characteristics of an ideal vaccine. In the edible vaccine, transgenic plants are used as vaccine production systems. The genes encoding antigens of bacterial and viral pathogens can be expressed in plants in a form in which they retain the native immunogenic properties. That means when you are producing a vaccine in a transgenic plant, you are introducing the changes through the genes, and thereby the newly produced plants will have the immunogenic properties towards that particular antigen. Initially, this was thought to be a useful one for preventing the infectious diseases, and it is also found to have its application in the prevention of autoimmune diseases in birth control and cancer therapy, etc. The edible vaccines are currently being developed for a number of diseases in humans and animals. You know, the father of medicine, Hippocrates, said, "Let thy food be thy medicine." So, whatever the food we eat, that should be the medicine for the disease which is going to be caused in the human beings or in animals. So, according to this, the edible vaccines productions are coming into existence and playing a vital role. Now, why you want edible vaccine? Or otherwise, what are the advantages of these edible vaccines? These edible vaccines are needle free. That means, by the word edible means it is taken by mouth, that is by oral administration. So the oral vaccines provide mucosal immunity at various sites by secreting the antibodies. Later we are going to see the mechanism of these edible vaccines. Then you will understand the fact. No need to worry about the reuse or misuse and lack of sterilization. So that's why it leads to low risk of infections. And the next advantage of this edible vaccine is the economy, cheap. The production cost is very, very cheap. For example, the estimated cost of growing an antigen for one dose of hepatitis B vaccine in an unprocessed form is approximately $0.005. So you can imagine how cheap the edible vaccines are. Administering oral vaccines would require a little or no training at all. So here we are reducing the cost of the technical people also. The next advantage is storage. Usually when you see your vaccine sera or toxoids, usually they are stored in the refrigerating conditions. But here in case of edible vaccines, they are heat stable, so they do not require cold chain maintenance. If the local or native crop of a particular area is engineered to produce the vaccine, then the need for transportation, distribution also can be eliminated. And next advantage of this edible vaccine is safe. The use of edible vaccines are safe. Most importantly, they trigger the immunity at the mucosal surfaces such as mouth, which is the body's first line of defense. And next, they require no purification. So edible vaccine activates both mucosal and systemic immunity. So these are all the various advantages of this edible vaccine. So now we will see how these edible vaccines are acting in the body. Otherwise, the mechanism of action of edible vaccines. The goal of oral vaccine is to stimulate the mucosal and systemic immunity against a pathogen. So as I already told you, edible vaccine means it is to be taken orally or by mouth. So when you just take the edible vaccine orally, so as soon as we take the food into the mouth, we are masticating it. Likewise, the edible vaccine also undergoes mastication process. Then it goes to the stomach and then into the intestine. When it goes to the intestine, the plant cell degradation occurs. As a result of this, by the action of digestive and bacterial enzymes. That means when you take the edible vaccine orally, it goes on through the GIT and reaches the intestine, where the cell degradation takes place by the digestive enzymes and also the bacterial enzymes. And next, what happens? The Peyer's patches, which are present on the mucosa of the intestine, they are the enriched source of immunoglobulin A producing plasma cells. And these patches will have the potential to populate the mucosal tissue and serve as a mucosal immune effector site. The breakdown of edible vaccine near the Peyer's patch, where around 30 to 40 lymphoid nodules are present on the surface of the intestine. And these nodules, lymphoid nodules, they contain numerous follicles. These follicles, they act as a site from which the antigen penetrates the intestinal epithelium and thereby accumulating antigen within the organized lymphoid structure. Where these lymphoid structures are present, they are present on the mucosa of the intestine. So what happens there? When these edible vaccines are broken down near to the Peyer's patch, where the lymphoid nodules are present in which the follicles are located. So once they get triggered, the antigen penetrates. That means the antigen penetrates into the intestinal epithelium and thereby it starts producing the antibodies. How they produce the antibodies? Let us see now. The antigen comes in contact with the M cell, and this M cell passes the antigen to macrophages and beta cells. These beta cells activate the T cell, that is the helper T cells, to provide immune response. So by this way, the immunity is activated by the edible vaccine. So here we can see the pictorial representation here. The antigen from vaccine, that is when the edible vaccines are degraded by the intestinal enzymes and bacterial enzymes, the antigen comes over here. So as soon as that is in the lymphoid nodules, the follicles, and it is penetrating through the epithelium and enters into the M cell, and from this M cell the antigens are passed on to the one way macrophages and in the other way to the beta cells. And what happens? These macrophages along with the T helper cell stimulates the production of the antibodies. Likewise, the B cells along with the T helper cells, they produce the antibody and it is released. Okay, so here you can see the stepwise mechanism of action. The M cell passes the antigen to the macrophages and beta cells. This is the M cell, this is macrophage, and this is the beta cell. And second one, the macrophages display pieces of the antigen to the helper cells from here to here. And third, the T cells stimulates the beta cells and seek out antigens at the distant sites. And then this beta cells produces the antibodies and they go into the circulation which will neutralize the antigen. Understood? This is the complete mechanism of action, initial response. So, and then here the virus is arriving here, and this one, and one side it is making the cells getting infections, and in the other way it enters into the beta cell, sorry, M cell, and this M cell transfers the antigen to the macrophages and also to the B cells with the help of the T helper cells. Again, the antibodies are produced and they are neutralized by the invaders. Okay, so this is a clear pictorial representation of the mechanism of actions of edible vaccines. And next, the development of edible vaccine. So how will you develop or how will you produce an edible vaccine? Edible vaccine can be produced by two ways. One is by introducing the entire structural gene into the plant by transformation vector between 5 and 3 regulatory element. So when you are introducing or inserting an entire structural gene, this will allow the transcription and accumulation of encoding sequence in the plant, and they produce the transgenic plant with the edible vaccine. In the second case, you are not inserting an entire structural gene. Here, the epitope within the antigen are identified. That means a particular codon. That means, in other words, the DNA fragment of encoding, these can be used to construct a gene by fusion with a coat of protein gene from a plant virus. That is by using the Agrobacterium tumefaciens, where the gene transfer is taking place by using the vector, plant vector. Okay, so edible vaccine can be developed by two ways. One is by inserting the entire structural gene and then transforming the vector between the segments, regulatory elements, getting the transgenic plant. And another one is a particular codon or DNA fragment is encoded by using the plasmids, using the host and the vector. So here very clearly you can see the production of the transgenic plant. So this is the antigen from a pathogenic organism. This is a complete structural gene where this is the TMV, that is a vector. The structural gene is encoded over here. The plant transformation vector for a foreign gene expression, and that is taking place or mediated in Agrobacterium tumefaciens. The genetic transformation is taken place, and this is allowed to act on the callus removed from a plant, and that is developed into a whole transgenic plant, from where you will get the leaves, which is concentrated with the antigens, or the potatoes, which is used as a edible vaccine. And from the leaves also the foreign antigen can be produced, or from the potatoes itself as an edible vaccine it can be used. So this is one way the entire structural gene is transformed to the plant through the gene transfer mechanism by using the vector Agrobacterium tumefaciens. And the other way is, instead of transferring the entire structural gene, a DNA fragment encoding the epitope is transferred to a chimeric gene, and then the infection of the plant with the modified virus, and finally from the plant the infected viral antigen can be isolated. So this is the production of edible vaccine antigen in the plant tissue. And what are the different methods of transfer of fragmented DNA or gene into a plant? One is, as I already told you, plasmid vector carrier system. That is by using the vector Agrobacterium tumefaciens method. And next one is microprojectile bombardment method. That means by using an electrical gun to the target organ directly, the gene will be transferred by using the microprojectile bombardment method. And next one is electroporation method by using the differences in the ionic changes. That is like nothing like your electrophoresis method. Okay. So these are the three ways by which the gene can, or a fragment of DNA can be transferred. So this is the transfer of gene by using a vector Agrobacterium tumefaciens and developing a whole intact plant with a edible vaccine. So here you can see a bacterial gene. This is a vector where it contains the antigen gene, a plasmid, and the antibiotic resistance gene. And here the plant cell, that is nothing but the a part, the explant from the leaf Pramodia. And what does it contains? It contains the gene as well, and also you have genes for the area for the gene transfer, and also it consists of the DNA. And this, what happens here, the expose, what we have to do, we have to select an explant, that is a leaf. It should be exposed to the bacteria which carries an antigen gene and an antibiotic resistant gene. And this, what happens, we have to culture this leaf so that the bacteria will deliver the antigenic gene into the leaf cell. And then what you have to do, you have to see this leaf should be exposed to an antibiotic solution. Why we have to expose? To kill the cells which causes the diseases. And you have to wait for a time that some of the cells will survive and they will multiply to form a callus. So where you have seen the formation of callus which are resistant to that antibiotic solution, and they are growing in the medium. And then this callus should be grown on a medium to sprout and to give out the shoots and the roots. And finally, this transgenic plant can be transferred to the soil where the plantlets can be produced, and from that plantlets the edible potato vaccine can be developed or can be obtained. So this is the way how the gene transfer is done by using the vector Agrobacterium tumefaciens. Okay. And here there are the examples of the plants where the different plants were used for the production of the edible vaccines like tobacco, potato, banana, tomato, rice, lettuce, soya bean, alfalfa, muskmelon, carrot, peanuts, wheat, corn. So these are all the various plants which are used for the production of the edible vaccine. And here you can see the different plant parts and what are their advantages and what are the disadvantages it is produced or facing. You can see the tobacco plant is used for the production of edible vaccine, and where you can see the advantages. It is a very good model for evaluating the recombinant proteins, low cost of preserving systems, and easy purification methods, and large harvest a number of times in a year can be done. And the disadvantages is it produces toxic substance along with the required edible vaccine. And next, in case of the potato, we can get dominated clinical trials, easily manipulations and transformations can be done, and it can be propagated from its eyes stored for a longer periods without refrigeration, and it needs cooking. So during cooking, denaturation of antigen can be taken place, and thereby decreasing the immunogenicity. That is one disadvantage, major disadvantages in case of potato edible vaccine. And next one, banana, no need of cooking over here, so proteins are not destroyed by cooking, so trees take around 2 to 3 years to mature. This is the disadvantage. And transformed trees take about 12 months to bear fruit. And again, the advantages in banana, if you see, it is inexpensive, grown widely in developing countries. Okay. And it contains very little proteins, and so rapidly it gets spoiled after the ripening. Okay. And large amounts of recombinant proteins can be produced. And next, tomato, it grows quickly and cultivated broadly in nature, higher content of vitamin A, so immune system can be boosted like anything, and they are heat stable, and antigen containing powders can be made out of it. So it is made into capsule and then it can be given. And the main disadvantages is within few days the ripened fruits get spoiled out. And rice, it is a commonly used baby food because of low allergenic potential and high expression of proteins and antigens can be done. And again, here also it requires a specialized glass house for its growing conditions. And lettuce, fast growing, direct consumption and spoils readily. Soya bean, large harvest, number of times in a year we can do the cultivation with soya bean and alfalfa. That is the advantages. And with muskmelon, it is fast growing and easily propagated by the seed and easily transformed. So these are all the advantages of the various plant or fruits in the production of edible vaccines, and also they face little of the disadvantages too. And here there is the plant viral system patents. That is, you can see what are the different viruses and what are the targets, the host, and the plants used for the production of edible vaccines towards these virus, and what is the mode of administration over here. and they have made the patent of these things. Okay, so that is enterotoxigenic E. coli, E-tech. It is the target host is human. That plant used for the production of vaccine is tobacco, and mode of administration is oral. Most of the things they are oral, and some of the things are by the injections. Okay, especially the Vibrio cholerae oral, and hepatitis B virus through humans. It can be produced by various like the potato, tobacco, lettuce. Okay, and then the Norwalk virus, that is the polio causing one, that is tobacco and potato are the plant used to produce the edible vaccines, and usually they are taken by the oral route. Okay, so these are the some of the examples of viruses which are grown on the different types of the plants, and the targeted uh, host like the humans, and or sometimes with the animals, and with the mode of administration. And next we will see about the current status of this edible vaccines, the clinical trials. So these are all the extra informations what you can know about the edible vaccines. If you see, this is enterotoxic E. coli. It is developed by Boyce Thompson Institute in USA. It has accomplished the first published successful human trial in 1997, and 11 volunteers were fed with this transgenic potatoes expressing this. Out of this, 91, that is 10 people, the individuals, they developed the neutralizing antibodies, and 6 developed a mucosal response. Okay, and next one, the clinical trial of Norwalk virus, that is against the polio. The transgenic potato was used to express Norwalk virus antigen, and 19 out of 20 people fed with the transgenic potato expressed Norwalk virus antigen showed seroconversion. And attempts are underway to engineer bananas and powdered tomatoes to express the Norwalk virus. And cholera virus, if you see, the transgenic potatoes were expressed with Vibrio cholerae and shown to be effective in mice. Eating one potato a week for a month with periodic boosters was said to provide immunity in this case. And measles, mice fed with tobacco expressing MEH could attain antibody titers 5 times the level considered protective for humans. So MEH edible vaccines will not produce a typical measles. Little difference between that. Transgenic rice, lettuce, and baby fed food against measles are being developed. And for the hepatitis, the parenteral VLPs could invoke specific antibodies in mice. The first human trials of potato-based vaccine against hepatitis B have been reported the encouraging results. And the amount of HPS-AG needed for one dose could be achieved in a single potato. When it is cloned into CAMV or plasmid HPS-AG, subtype showed higher expressions in roots as compared to the leaves of the transgenic potato. And this is the current status of plant-made vaccines for veterinary process. This is the pie diagram. And here you can see usually the Newcastle disease, which is a very common disease affected to the poultry and wild birds. So NDV transmission occurs through the direct contact with the secretions or discharge of infected birds and contact with fomites. So the world's first regulatory approval for PMV was against NDV. The NH protein from NDV was expressed in tobacco cell system and found to retain the size and immunoreactivity. And next one, foot and mouth diseases in uh, cattle. The causative pathogen is a FMD virus. This FMD virus is a single-stranded virus positive sense RNA virus having 4 caspits, VP1, VP2, VP3 and VP4. The VP1 protein caspid is a critical determinant for the vaccination against this foot and mouth disease. And the studies have shown the potential of using VP1 caspid protein as a subunit PMV candidate in potato, tobacco and tomatoes. And next, avian influenza. There are 3 types of viral influenza in birds like A, B, C with distinct pathogenicity and genome properties. Influenza type A virus is endemic in birds. It is contagious not only to avian species but also to the variety of mammals. And the types B and C infect mainly humans, but they are less lethal. And high accumulation of VLPs made from HA antigen was observed to be immunogenic. And mice immunized with intramuscular doses of purified H5 and VLPs were protected against influenza virus. So these are all the different clinical trials and the edible vaccines against the animals are also under trials and are having a good response as an edible vaccine towards the particular diseases. So what are the safety issues in these edible vaccines? If you see, sometimes they are allergenic and toxic potential of plant components, especially glycans and nicotine, and their potential for interference, production of oral tolerance, risk of a typical measles in plants with cloned measles virus, health and environmental risks of GMO, and prevention of misuse and overuse. And the public perceptions and attitudes towards the genetic modification is not acceptable 100%. So that also will have some limitations in the production of this edible vaccines and getting the license with the quality controls. So that is about the edible vaccines. Thank you.